Hello everyone, welcome to Glecta, and this is our first GCSE live, and this is for your subject biology. I hope you guys are very excited. Okay, hello Dibas, hello Mothership. How are you guys? Are you guys excited for the biology session today? I hope you are, because I am. Okay, so for whoever is here for the first time, let's have an introduction about what Glecta is. So Glecta is a London-based tutoring platform which provides tuitions for 11 plus, KS3, GCSEs and A-levels for all the major boards. So if you guys have not still enrolled with us, you may, and I think you should. Now coming on to what we're going to cover today, we're going to talk about the major topics of biology that are there for GCSEs. Although we all know we can't cover all the topics in one class because that's too vast of a syllabus, but I have come up with some that are the most important ones. I would say a part of most important ones. So today we're going to discuss about the DNA, protein synthesis, digestive system and enzymes. So I hope you guys are ready for the session. Okay. Hey. Okay, Mothership says I love biology. I do too. That's same. Okay, Debas says this is Soma Robin. Hello. Okay. So moving on to the first thing. Now here on the screen you see a question that says, where on the diagrams should these labels go? So take your time and think about it, that if you have this diagram, how are you going to label it with the following words? It says chromosomes, nucleus, DNA and genes. So focus on the screen, think about it, what should be the nucleus? Do you guys know what the nucleus is? Quickly tell me everyone, how do you think it's gonna be and where do you have to put the nucleus? What do you think is the chromosome? DNA and genes. Anyone? Okay, nobody knows this? Should I think of it that way? Okay. Okay, if not this way, then try it this way. Can you put them in the order of their sizes from the smallest one to the largest one? Can you do that? Anyone? Okay. Okay, yes, that is correct. The part of a cell is a nucleus. So which means if you see a cell, in the middle of it, a part of it should be the nucleus. Vijay says that it is the center part of the cell. That is correct, Vijay, very good. So yes, if you have this diagram and you have to label it, you would go chromosome, which is the section in which you see a lot of rolling over. Then you have the genes, there's the DNA, and then we have the nucleus, the center of the cell. And now if you have to label them from an order of their sizes, the smallest one is the gene, then the second one would be DNA, chromosome is the third one, and the nucleus is the fourth one. So this is how you can go from the genes to the DNA. Now coming to, why is it that DNA is not the smallest one while gene is? So let's talk about what each of them is. So let me ask you a question first. What do you think there's the difference between chromosome and the DNA is? Okay, nucleus is the center of the cell, contains DNA. That's correct. Double strand, helix strand is DNA. That's correct, Tanishka. And okay, nucleus, chromosome, DNA, genes. That is correct, Mothership. Double helix structure, that was DNA. That is correct. Very well done. And can somebody now tell me the difference between the chromosome and a DNA? We all know what a DNA is. We always use it in our daily life. We say it's not in my DNA. It's not on his DNA, her DNA. So we talk about DNA a lot. So Dennis says deoxyribonucleic acid. That is correct. DNA is the deoxyribonucleic acid. Then what is chromosome? Okay, so confusion, a lot of that. To clear that out, if this thread, this entire thread here on the screen is the DNA, which means if you zoom this up from any part, you may see a double helix that you said is the DNA. And DNA is quite literally in meters, and I mean meters. So I'm not talking about 1 meter, I'm talking about more than 1 meter. And because DNA is so long, and I have to fit that long thing into a cell, which is microscopic, I can't see it with my own eyes, I need a microscope for that. And then I have to put it in the nucleus of that cell, which is all the more even smaller, and hence I need to compress this. So how do I compress? I roll this thread over and over and over again to make a chromosome. So you can pretty much say that a wrapped up version of DNA is a chromosome. I hope this clears the concept between what is a DNA and what is a chromosome. Now, for the third thing, what is a gene? Gene is what is you, what you are made up of. You're everything that your genes are. You have a gene for eye color, you have another one for skin color, you have another one for your hair color, what should be your height, how should be the texture of your hair, how should you have enzymes in the body, what proteins you have in the body. Everything that you're made up of from head to toe is your genes. And these genes are passed down from our parents, which means if somebody says your eyes look like your father's, or somebody says your eyes look like your mother's, they are talking about that the genes are the same. Whatever genes your parents, your father had for the eye, you have the same. So this is the difference between genes, DNA and chromosome. And we all know what the nucleus is. It's the center part of the cell which contains the nucleus, which controls everything in the cell. Now I hope this is clear to all. I hope nobody is now confused with the 3 alphabets and the 3 words. Now moving on to this next thing, which is this. Now here on the screen what you see is chromosomes. Chromosomes that are arranged in a pattern. And this is known as a karyotype. Anybody can have their karyotype done. All you have to do is go to the doctor, say that I need to get my karyotyping done. And you can have. But what is this karyotype? This karyotype is your chromosomes, all the chromosomes in your cells, they are arranged. So it's a very systematic version of it. Although in the cell you might not have it like that, you may have them random. But these are the chromosomes that are arranged from top to bottom. Now talking about how many chromosomes do you have in a cell? How many chromosomes do you think we have in the cell? Okay, mothership says X and Y defines our gender. That is correct. For example, if it's XY here, that should be a male. And if it is XX, that should be a female. So that is correct. But then what is your number of chromosomes in a cell? Anyone? Millions. No, we have numbered. Tanishka says 23 pairs. That is correct, Tanishka. We have 23 pairs of chromosome in a cell. And when I say a cell, I mean a body cell. Which means from head to toe, all the cells in the body have 46 chromosomes in the pair of 23. So in 23 pairs, for example, here you see pair number 1 having 2 chromosomes, pair number 2, pair number 3, pair number 4, and like this you have 23 pair of chromosome, which means in total the number of chromosome is 46. Now, there is an exception to this though. There are cells in the body that only have 23 chromosomes. Which one do you think is that? Which chromosome would have, which cell, instead of having 46 chromosomes, only has 23? Okay, 23 pair, that's 46, that's correct. And which cell has 23 chromosomes and that's it? Nobody knows about that? Sex cells. No mothership, it is sex cells, what we call as gametes. Gametes are what we are made up of. So a cell from the mother, a cell from the father comes, and then they fuse together to make a zygote. In those cells, the gametes, the sex cells, they have 23 chromosomes. And how do you think that is sufficient? Do you think it is sufficient? Is it okay to have only 23 chromosomes in a cell? How do you think they work then? Because I just said DNA has genes. Don't you think having 23 chromosomes would make you have less genes? Then how does the baby have all the genes? Think about it. Quickly, everyone. I want answers here. Quickly. Okay, some kids say stems, gametes. Okay. And have you guys liked the video? If you guys are watching this and you still haven't liked the video, like, share and subscribe, because we are going to, in the middle of the session somewhere, I am going to announce a kid who was most indulged in the class. So you should do that, answer your questions properly, and maybe next time you're going to be that lucky kid. Okay, 23 from the mother, 23 from the father. Exactly, Tanishka, very good. That is true. Sex cells or gametes only need 23 chromosomes because the 23 comes from the father, 23 from the mother, making a 46 in the baby, which means the baby is normal. The baby has all the genes that they need. So the zygote has 46. And hence, sex cells and gametes are the only cells in the body, the entire body, that has 23 chromosomes. If somebody does not know the names of the gametes or the sex cells, I should tell you it is sperms in males and egg cells in females. If you hear that word ever, do not get confused. They are talking about the sex cells itself. Down syndrome is where you have a partial or full copy of chromosome number 21. That is true. And that's why karyotyping is very important. Because just looking at this, for example, if I see somebody's karyotype like this, I would say this is a normal person. Male or female, but a normal person. But if I ever have an extra chromosome here, that's Down syndrome. So the kid would be born with abilities that are different from others. So they would have a Down syndrome, they would have some learning disabilities sometimes. We do have a triploidy of 18 chromosome as well, and so do we have for 13. So these chromosomes can be copied, and because of that, the kid or the person may be born with some kind of disabilities. So that is true. Very good, Dennis. Now, moving on to the DNA and what is it made up of? What is the structure of the DNA? Okay, so we all know DNA like this, a double helix with strands in the middle. Let's try to break this. So to break this, what I'm going to do is unwind it. So when I unwind it, it looks like this. Pretty much like a ladder. And if now I break this ladder, it would be like this. Which means technically DNA is made up of whatever this slant D is made up of, because that's how we can break the DNA. this slant T here is what is known as a nucleotide. So DNA is a polymer, it says. Polymer means having something repeatedly for n number of times. So for example, if you have a lot of pencils, that would be a polymer, having so much of something. So repetitive unit of something that's attached together is a polymer. And DNA is a polymer, but it's a polymer of nucleotide. So a lot of nucleotides together are known as DNA. And they all have three things. They have a sugar, which is pentose. Pentose means it has 5 corners, pen, 5. Then we have a nitrogenous base. We're going to talk about this in a bit. And then we have a phosphate group. So we have three things: sugar, phosphate, and the nitrogenous base. Sugar makes all the difference. Sugar can be for deoxyribose, which is a DNA, but it also can be a ribose, making it an RNA. So pretty much a possibility of having all the kinds of sugar, but these two make my nucleotides. So again, if it's ribose, it's going to be an RNA, and if it's deoxyribose, it's going to be a DNA. These three things would make my DNA. You guys are awfully quiet in the comments section. What happened? Do you guys understand everything that we're talking about? If you guys have questions that you guys would like to ask, you may ask them in the comments section. And also in the comments section, you have a free mock test for your GCSE's biology. So you may go to that. It would be there, and if you can't find it in the comment section, at the top of it, it's been pinned for you. So go there, download your mock test, and try doing that at home as well. So for that exact reason, I am telling you the topics so that you know that these are the important ones. I may get some questions from this. So you should focus on the class, but keep commenting in the chat section so I know you're present, and download the mock test. Now moving on to what I said, the nitrogenous base that I'm going to talk about in a bit. We have 4 types of nitrogenous bases. They are adenine, thymine, cytosine, and guanine. And I'm talking about when I have a DNA. So in the DNA, we have deoxyribose sugar, and then we have 4 different types of nitrogenous bases. Now nitrogenous bases are very, very specific, and for a fact, the kind of nitrogenous bases you have in your body, let's say it's adenine, thymine, guanine, the kind of arrangement they have. For example, first I have adenine, then thymine, then guanine, then again adenine. So if I have a pattern like this, it would define my body, because this may be coding for my eye color. So the pattern, the codes of the DNA are the nitrogenous bases. Whatever the nitrogenous bases are attached to, and in whichever pattern they're attached, that makes the difference. Now, coming to how do I make that ladder? Because I talked about nucleotide being this T. Let's talk about the other T and how they're attached. So the nitrogenous bases can connect to each other. They can form bonds with each other. And what kind of bonds? Hydrogen bonds. So adenine is attached to thymine by a double bond, and guanine is attached to cytosine by a triple bond. So these are hydrogen bonds. Don't forget this, because this question and this knowledge would help you in your GCSEs regarding the questions that may be related to DNA replication as well. Once this is clear, now if I have to review everything, what I had till now is that DNA is a polymer. DNA is made up of 3 things: deoxyribose sugar, which is also known as a pentose sugar, a phosphate group, and a nitrogenous base. Now, the second thing that we learned is that there are nitrogenous bases of 4 different kinds: adenine, thymine, guanine and cytosine, and they are attached to each other. Now if you look at this here, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, and they're attached to each other. Here as well, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. They're attached to each other, which means technically the strands of the DNA is phosphate and sugar and phosphate and sugar. And then in the middle, the rungs of the ladder are made by nitrogenous bases and their bonds. Once you have these bonds, you have a structure like this. And here if you see, it says sugar phosphate backbones. That's what we call it, pretty much. So sugar, phosphate and nitrogenous bases, that is correct, Mothership. ATGC, that is correct, Vijay. So yes, talking about the DNA, we have backbones made up of sugar and phosphate. So we call them sugar and phosphate backbones. And then we have a turn for them. So when they turn, they make the double helix structure of the DNA that we know of today. This is the structure of DNA. I hope everyone is very, very clear with this, and I hope you guys now understand the structure of DNA, because this would give you a lot of knowledge about how protein synthesis work as well. Now, this wraps up everything that we've talked about the DNA so far, which is the phosphate and the sugar section of the nucleotides form the backbone of the DNA strands, which is the sides of the ladder, and the base pairs of each strand connect to form the rungs of that ladder. So in the middle, they're connected and then they twist. It is the sequence of bases, which is A, T, G and C, that holds the code for the formation of proteins. And those proteins would do everything in your body. They would give you a specific eye color, they would give you a specific height, a specific hair color, a specific skin color. So everything in your body would be actually defined by the kind of genes you have, the kinds of codes you have, and how they're able to make a protein out of that. Now moving on to that, in the last session, like I said, we are always going to be announcing the winners for the most active kid in the class and the most interactive one as well. So keep interacting with me, keep on answering the questions on time, and maybe the next time you're going to be the kid. Now moving on to from DNA to protein. Now technically, if you think about it, we know that the DNA is present in the nucleus, and we know that the DNA can't leave the nucleus. It's pretty much bound to that place because nucleus has a membrane to itself through which the DNA can't come out. But that's because DNA is double helix like this. So if I am able to somehow get the codes of the DNA copied onto something that's not double helix and it's not that big, maybe I would be able to get those codes out from the nucleus. And then once I get them out of the nucleus, I can make proteins out of those. And whatever my proteins say, my body would do. This is the central dogma of our body. Everything in our body works according to this. So what happens is, first your DNA keeps on replicating. In every cell cycle, it will replicate so that it can make more and more DNA, so that every time a cell grows and divides, the cell gets the DNA. So for that, it has to keep on replicating its DNA. So it would do that. Then once I have a lot of DNA, I am going to make RNA out of it. So RNA is basically like a messenger of that entire process. Why? Because I have to go from DNA to proteins, and I have to get the DNA codes out from the nucleus. So this process in which RNA is made, we call this process as transcription. Why do you think it's called transcription? Have you guys ever heard the word transcribe? When we have codes for something, instead of having the actual words, instead of saying, let's say, you know, when we have codes for different kind of things like army people do, so you have to transcribe that. So whatever you want to say, you transcribe it and forward it. So that's what transcription is. It's going to get the codes from the DNA. Once it is transcribed, then you can translate them. So it's called translation when the RNA is converted into proteins. Pretty simple. And because if you think about it, I had to go from DNA to proteins. RNA is in the middle, working out for the same process. We can pretty much call this RNA as messenger RNA. And that's what this one is called. It is called the messenger RNA because it's trying to get the codes from the DNA so that they can come out and make proteins for your body. So this was about the DNA. Now I have a few facts for you guys to read about the DNA. So I'm going to get my face out of the screen so that you can read it properly. And I hope you guys are ready for some facts. There you go. So it says, our closest relatives are chimpanzees and bonobos. We have more than 98% DNA in common with them. Do you guys know that whenever you have to compare between, let's say, humans and chimpanzees and bonobos, instead of comparing adults, like an adult bonobo monkey or a chimpanzee with humans, compare the babies. Look at how similar the babies look. And that's where you would understand that we do have some relatives out there because the babies look very similar to that of humans. Every human shares approximately 99.99% of their DNA with the other human, which means I am 99% similar, 99.99% similar to you, even when we're not relatives. That's because we technically have the nose at the same place, eyes at the same place, hair growing at the same places, lips at the same position, the chin, the neck, the shoulders. The body structure, what we're made up of, the organs, the bones, we're technically the same. We just have variations in that. It's different skin colors, different heights, different weights, different hair. So all that may differ, but technically we're the same. The entire human DNA sequence is called a genome. There are 3 billion base pairs in human genome. Well, that's quite a lot, if you ask me. But a genome, for those who don't know, is actually the collection of all the DNA that we have. So, for example, if I'm talking about my genome would be all the DNA, all the chromosomes present in one cell. So that would be my genome. So that's how much genome we have. Now the next one, if you type at top speed, which is 60 words per minute, for 8 hours a day, it would take you 50 years to type the complete human genome. That is a lot. Like, I don't think it's possible for somebody to do in one sit and go, but probably someone should try. And the last one is, the words for "it's a small world" would have been encoded in artificial DNA and placed in the host bacteria resistant to most conditions. Thankfully, this is not the only legacy we are leaving though. So what they're trying to say is that they're actually trying to encode a message and put it into bacteria that are resistant, which means those bacteria are not going to die. So I hope you guys enjoyed these facts and I hope you learned something new. And let's move on. Now talking about the protein synthesis. Now I don't want to say a lot about protein synthesis because I'm going to show you guys a video so that you guys understand this. But let me tell you the basics. So what happens is, I had the DNA, I got it converted into the mRNA. Now the mRNA is out. We all know from the first chapter of GCSEs that proteins are made in the ribosome, because ribosome is the site of protein synthesis. So we know that this mRNA have to go to the ribosomes so that we can start the process of making proteins. So what is going to happen is, once the mRNA reaches the ribosome, the process would start. Now ribosomes are quite simple. There's a huge ribosome, then there's a tiny ribosome, and they're connected to each other like this. In the middle, they have a little space for the mRNA to come. So like this, in the middle, we have mRNA coming up. Once we have the mRNA, I am going to start the conversion of those codes on the mRNA into proteins. So it's pretty much like a machine. Read one code, move forward, read one code, move forward. So look at this. Whatever code would be here would be read, and then a protein, or let's say amino acid, would come and join. Then the next code would be read, and the next amino acid would be attached. Then the mRNA would move forward, and then again amino acids would read the codes are read, the amino acid come, and like this the chain is formed and released from the ribosome. Hello Ramit. Okay, now let's move on to the video of protein synthesis. Look at this very carefully so you understand the entire process in one go. So this is the process of making the mRNA. Once you've made the mRNA, you can get that mRNA to the ribosome. Then the tRNA would come, and this is how the amino acid gets attached to each other to make a chain of proteins. Now protein is in a strand. DNA can be replicated to form the same human again. It must be replicated, but yes, that would be clones. An entirely different process, but human clones have not been made yet, but there's some researches going on the matter. So yes, so now this, talking about this video, if we go from the start, what happened? RNA was being made, so this is how the RNA is made. Once we have the RNA, this strand, the upper one, the blue one, would go to the ribosomes. So now we can go to the ribosomes. These are ribosomes, and in the bottom we have the mRNA, and these yellow, red, green, blue, these are the codes. These are A, G, T, C, the codes that we had, the base codes that we had. Then comes the tRNA. We did not talk about the tRNA. So tRNA is actually like, it's transferring the amino acids from your body to the ribosome so that we can make a chain of proteins. So here, a point to note is that proteins are made up of amino acids. So tRNA, the transfer one, is going to bring me amino acids, because as soon as it would read the code, look at this, it would pair up, it would get the code. Once it does, whatever code it is, it would get me an amino acid for that code. So for example, I have AUG, let's say. Now in RNA, we have U instead of T, so AUG. AUG codes for methionine, that's the amino acid's name. So if they read AUG, they'll get me a methionine. So like that, the tRNA gets me the amino acid that I need according to the codes that are read. So now playing the video again, the more codes it read, the more amino acid the tRNA is going to bring up, and the more amino acid it brings, the more connection it's going to make with them. And this, a chain of amino acid, is what we call a protein. And it can convert into a particular shape for its own function, and then it can form different kind of structures in the body and the different kind of hormones, enzymes in the bodies. So we have a lot of function for proteins. For example, all enzymes are proteins. We've talked about that a lot of times. So all enzymes are proteins. Then all hormones are majorly proteins. Then we have structural proteins. You see how your face is uplifted like this? This is because of proteins that are there in the face. So we have a lot of proteins that keep our face uptight and in the structure. Similarly, our muscles, they're held together by muscles that have proteins. So proteins make us. Now again, 3 types of proteins. We have enzymes, we have hormones, and then we have structural proteins that make the body. Are you guys clear on this one now? So this was simple enough for you now? Now we've talked about DNA and we have talked about protein synthesis. Shall we move on to the questions now? Is everyone ready here? Mothership, Eva, Bella, Emily, Ramit, Vijay, are you guys ready for the questions? Please say yes. Because I am pretty sure that if you've understood everything and you've heard everything very carefully, you will be able to answer. Okay, so let's start with the questions. Before that, like I said, I will give an award. So for the most interested kid and the most attentive kid in the class would get a free session with us. This would be the kid that has been talking the most in the class, in the comment section, interacting the most, giving the most amount of right answers, and just being active in general in the life, just being there attentively. And also, along with the free session, I want to tell you that if you've not enrolled with us, our new batches have already started, and if you are there and you're still not joined us, enroll with us today. For the details, you may see them at the bottom of the screen. So, and we also have some important contacts that have been sent in the comment section, so you can take them from there. Okay, moving on to the first question. Now these questions are from the past papers of GCSEs, so make sure when you're answering, you think about that. You think about the right kind of answers that may be there, which means you think about the scientific language you should use while answering the questions. Okay, so it says the nucleus of a cell contains DNA. We know that. Name the structure inside the cell nucleus that contains the DNA. Now I just talked about this and I hope you guys know this. So quickly everyone, try and tell me this. I'm going to wait for you guys now. So quickly tell me, what do you think is going to be the structure that contains the DNA inside the nucleus? Okay. Hazel, Mothership, Dennis, Vijay. Tell me quickly. I know you guys know that. It's easy. Chromosomes. Michael says chromosomes. That is correct, Michael. So you should say chromosomes, and sometimes genes are allowed, alleles are allowed, and even chromatids are allowed. So they always tell you the extent to which you can write, and that is correct. You can write chromosomes, chromatids, genes, or alleles. Pretty much the same things. Moving on to the next one, which is figure 1 shows part of DNA molecule. They do here. Name the part of the DNA molecule that has been labeled X. Tanishka says chromosomes, Ramit says chromosome, Hazel says chromatin, Noel says chromosome, Mothership says chromosomes, Dennis chromosome. Yes, the last one was still chromosomes. And let's move on to this one, which says name the part of the DNA that has been labeled X. We've learned about this just now. So quickly tell me, what is this pentagon right in the middle? Anyone? What's the pentagon in the middle? Pentose sugar. Well, yes, Mothership, it is pentose sugar. Michael says sugar. Yes, that is correct. Not the bases, Mothership, but the pentose sugar. So you can say sugar, you can say deoxyribose, you can say pentose sugar, but you can't say ribose, because we know that this structure is DNA and not RNA, so you can't say it's a ribose sugar. That would be wrong. Pentose base sugar, pentose, okay, pentose sugar, yes, that is correct everyone. So it is the pentose sugar. So don't say ribose. If you have to name the sugar for some reason, say deoxyribose sugar. Don't say ribose, that would be wrong. Moving on to the next question, it says what type of structures are labeled, substances are labeled A, C, G, T in the figure 1. So this is your figure 1. What is A, G, T, C? What is that? Alexa says sugar, and we're done with the sugar. Moving on to, but yes, it is sugar, still sugar. Moving on to, what are these substances A, G, T, C? Everyone, what is A, G, T, C? And have you guys liked the video? Did you? You're watching it and I know you're liking it. So hit the like button so I know you liked it, and so that we can continue having more lives. So like, share and subscribe. Nitrogenous bases, that is correct, Mothership. Michael says bases, that is correct. But Michael, I would want you to be a little more specific. When you say bases, bases is correct. But for the safer side, because we are here saying that, you know, we have to be specific for the words we're using. So you should be specific that it's nitrogenous bases, lesser chance of losing marks. So bases would be correct, nitrogenous bases would be correct. And you can also say the names of AGTC, which is adenine, cytosine, guanine, and thymine. So you can say any of these. So Hazel Brown says proteins. No, darling, it's not proteins. They're bases. They're nitrogenous bases. Okay. Not amino acids either, which are proteins. Are different. Amino acids are different. These were about nitrogenous bases. Moving on to the long question. These are the ones that everyone is scared of, and these are the ones that actually, you know, like scares us. This is a 4-mark question. It says protein synthesis occur in different parts of a cell. Describe the process of protein synthesis. Now quickly, everyone who has ever had classes with me who understands how to answer the long question, quickly tell me only the points that you should be mentioning in this question. Everyone, this is for 4 marks, so you know the rules. You're at least supposed to give me 4 points. So I would like to start counting your points. So quickly start telling me the points. It occurs in 2 stages, transcription and translation. Okay, that is correct. Hazel? What else? Be specific, very, very specific. Come on, everyone. I know you know it. It was not that difficult. Now, you guys have watched the video, right? Just play that video in your head again. Think what you saw, and then explain what you saw. That's it. That's all you have to do. That's the entire point of watching videos. Because I know you guys remember videos more than anything. Okay. Moving on to nitrogenous bases. Okay, okay. Okay, so amino acids join together to make protein chain. Okay, it moves out of the cell. It moves out of the nucleus. That's correct. It includes 3 steps. That was something, but yes, okay, let's move on and let's keep it connected. So we were saying that we can talk about that we have a specific template, which is my mRNA. There are 3 bases that code for 1 amino acid, like I gave you an example of AUG. Then amino acids are added to a chain in a correct order according to the template order. For example, after AUG, if I have ATA, then first amino acid for these 2, then next amino acid for these 2. Or you can say that the chain folds up to form a 3D unique shape. So we can work with all of that, any 4 out of these, and you would get complete marks. So the trick here, so the trick to be, who said not yet, do you like the lesson? Okay, let me read the chat section here. Okay. So I'm facing some network issues. We all did for a second, I guess. We all did face that network issue for some reason. Okay, moving on to the next one. Did you guys understand how to answer this one? No, it's not over. Yes, it's not over. Who said not yet? To do you like this lesson? Okay. Now we're moving on to, so you guys did answer. Okay, very well. DNA is replicated and then transcribed into RNA. The RNA brings amino acid to join together and then translated into proteins. Very elaborative answer, I would say, for that one. So now you guys, this is how you answer the question. The trick to remember that you have to do it this way is always, and when I say always, I mean for all the time, you should do 3 things. First is introduce. So introduction is important. When I say introduction, I mean, for example, the line says protein synthesis occur in a different part of the cell. They're quite literally giving you a hint that you should talk about ribosome. So in the introduction and also in the introduction of protein synthesis, you say that protein synthesis takes place on ribosomes. So that's your introduction. Then you start with a body, which can be 2 points, 3 points at max, not more than that, in which you can talk about that we have a specific template, which is mRNA, which carries the code, and those codes specifically code for specific amino acid, and then we have different types of amino acid coming forward, forming a chain, and then you can at last conclude the topic saying that once it is made, then it can form a 3D shape. So always make sure that you have your structures very, very specific. You have a structure to your answer, and then you should use scientific language. For example, instead of saying proteins are made, synthesized would be a good word. That proteins are synthesized in the on the ribosomes. And learning a lot. So I hope this is all clear, and I hope you understood everything. Now, if you guys want to stay connected with us and you guys would like to know what is happening at Glecta and what event comes up next, you can always join us at Telegram, which is a group. You can join us on our YouTube channel where you're watching. Great time, and instead of just having that internet thing that we all had, other than that, I loved it. I had a great time with you. I hope you guys did too. So I hope to see you guys the next time. Bye-bye everyone. Take care.